Hello and welcome to BioBiz Buzz, a podcast on the science and business of biotech, pharma and medtech. I'm Cormac Sheridan, and this week we're taking a look at T-cell receptor cell therapies. These are cancer immunotherapies in which a patient's T-cells are isolated and engineered to recognize part of a specific protein on the surface of cancer cells. Once modified, these T-cells are reintroduced to the patient to seek and destroy cancer cells expressing the targeted protein fragment or epitope. While the concept is straightforward, TCR therapies are complex and involve multiple components. To learn more about how they work and about what they can do, I'm joined this week by Elisa Kiebach. She's the co-founder and chief technology officer of T-Knife Therapeutics, a German firm focused on T-cell receptor cell therapies. Elisa, you're very welcome to BioBiz Buzz.
Thanks very much for having me.
Maybe we should begin by discussing the company's very distinctive name. I've been aware of T-Knife Therapeutics for quite some time, but I've not had the chance to speak with you before now. And I've always found the name kind of arresting. For me, it sort of sums up images of a team of scientists conducting some form of open surgery on a T-cell, digging down into the cell's physiology to develop, an understanding of its functions, and maybe in the long run to better engineer T-cell therapies. So as the co-founder, you were there when the name was born. Are you T-cell surgeons?
I guess you may say so to some extent. The name actually comes from my scientific co-founder, who's Professor Thomas Blankenstein. He's a PI at the Max Delbrück Center and the Charité, which are, the academic institutions where our foundational technology platform was developed. And I guess for Thomas, it just symbolizes, a little bit like the precision of a surgeon's knife that cuts out the tumor very precisely while actually keeping the healthy tissue intact.
Yeah, a surgeon's knife that operates at the cellular level. Yeah, I like that. And obviously, CAR T therapies are maybe better known at this point, and they have similarities, but they also have differences. The CAR T therapies, you're limited to sort of proteins that are highly expressed on the surface of a cancer cell. And there are some reasonable options in haematological cancers, especially B-cell cancers, where the CD19 antigen is proven particularly attractive. But there are few, if any, antigens that are uniquely expressed by solid tumours. And we've had 7 CAR T approvals over the past 8 years, all of them in hematological cancers. But we've only had one TCR cell therapy approved, and that was only last year, Adaptimmune's Tecelera, that gained FDA approval last year for treating synovial sarcoma. Why have there been so few TCR cell therapy approvals, do you think?
Yeah, really good question. Well, I think let's start with conceptually CAR T and TCR-T are not that different. I think in both approaches, you really modify T cell, typically an autologous T cell from a patient, with another receptor that is specific for a tumor antigen, and then redirects these T cells to kill tumors. And the major difference really between TCR and CAR T lies in the type of antigen and that these receptors can target. TCRs are really unique in the sense that they can detect very small fragments of intracellular antigens that are then presented on the cell's surface and so-called MHC molecules, while, as you said, CARs are really limited to bind only cell surface molecules like CD19 or like BCMA. And now, white blood cells, the origin of liquid tumors, have very distinct surface markers, and CAR developers have, leveraged that for targeting heme tumors, which are derived from these white blood cells. And what we've seen, obviously, as a side effect is that the healthy blood cells, like B cells that are also CD19 positive, get eliminated as a side effect of the CAR T therapies. But that essentially is not a problem because, humans can live without B cells with some restrictions. I think while CARs have been really perfectly suited to treat heme tumors, TCR developers from the beginning on have really retreated more to focus on solid tumors, where solid tumors, the antigen landscape is very different because it's very hard to find solid tumor targets that are distinct from healthy tissue in contrast to B cells. And most of these antigens are, shared with vital organs like heart or like the brain. So, using TCRs really allows you to target a much broader range of intracellular antigens, and it broadens the space for actually safe solid tumor targets. And what I would add is that unfortunately with solid tumors, very different to heme tumors, we have a lot of additional barriers to the immune system. It took a lot of time for the field to really learn which solid tumor indications respond to T-cell therapy, which targets are safe enough, and also how to actually further engineer T-cells beyond just the CAR or TCR to protect the T-cells from the immunosuppressive tumor microenvironment. So what I would say is we not just had one TCR approval versus 7 CAR T approvals. I would say what we've seen is that there was an earlier success in heme, and that was mostly driven by CARs. being best suited for that. And what we now start to see is the first success in solid tumors, which is more challenging. And I would say remarkably, that first approved product in solid tumors is actually a TCR-T.
Okay, yeah, that's a fair way of putting it. And yourselves now, I mean, your lead investigational therapy is about to start a first in human trial. That's TK6302, and that targets the PRAME antigen, which is expressed, I understand, in a range of solid tumor indications. And maybe we can talk about that trial and the kind of patients it's going to enroll, the number of patients that will be involved, and the sort of main outcomes that you plan to measure.
Yeah, sure. Our ATLAS study is a first in human open label phase 1 clinical trial. And it will focus on patients with late stage metastatic solid tumors. And namely, these are ovarian cancer, uterine cancer, melanoma, squamous non-small cell lung cancer, and triple negative breast cancer. And all of these tumor types have in common that they really have a high unmet medical need. We've seen a lot of progress in the field, but still there remain patients with a high unmet medical need. And what we know is that additionally, these tumors have a very high prevalence of PRAME expression and, are just a logical choice for investigating a PRAME-directed TCR T-cell therapy. And this is with patients who have basically exhausted all available treatment options at that stage. For the ATLAS study, we plan to enroll up to 24 patients in a dose escalation part of the study before we would then move on to the dose expansion phase. And what I think is quite novel is that for the dose escalation, we're using a very a more novel approach for the dose escalation, which is an adaptive approach with a Bowen design, which is much more precise than a classical 3+3 design or identifying the optimal dose. Yeah. And from the outcomes, so the main outcome of the study will obviously be safety and also defining the optimal biological dose. But we, of course, are also very excited to look at the anti-tumor response and the set of biomarkers that we will measure.
Yeah. I mean, from a safety point of view, obviously, in a first in human trial, that's always going to be the kind of primary endpoint and concern. And both CAR T therapies and TCR cell therapies have, especially in the early days in the clinic, there were toxicities, some of them expected and some of them unexpected, that were arising. And from your point of view now, given where the field is at, what do you think are the potential toxicities that could arise during this trial, and how do you control them?
Yeah, so let's start with the clinical sites, right? So with the sites that we're working with are really centers of excellence and very experienced in treating patients with cell therapy. They're really experienced in managing toxicities. And the classical side effects that we're seeing with cell therapy are cytokine release syndrome, as an example, right? And I think the field has really moved on in understanding how we're managing cytokine release syndrome. And the physicians we're working with in particular have a lot of experience with that. And I mean, there's a classical path for that, right? You hospitalize the patients for the first 14 days. You can monitor them closely and then obviously do an intervention if need be. But what we're also learning is that in generally, TCR T cell therapies for solid tumors, in particular also for PRAME as a target, seem to be very well tolerated. So we're not particularly concerned when it comes to that. And I would also say what people sometimes forget, right? What's so exciting about cellular therapies is that we're talking about this one and done treatment, right? Patients get one hospitalization, one treatment. They're having a transient period of potential side effects, and then they're done. And they can leave the hospital. They can go back to lead a normal life. They can potentially go back to work. Whereas in a lot of other treatment regimes, you get like a continuous treatment where you need to return to your physician, every 3 weeks for it to stay on course. And I think that that's really exciting about cell therapies still.
Yeah. And PRAME, a term we've used quite a lot already. I mean, it's an acronym, and the full name is quite a mouthful: preferentially expressed antigen in melanoma. You really do have to ask yourself who gets to actually coin these names. It's so often the case in biology that a protein, it's named after its first identified function. But obviously we now know, and given the list of cancer indications that will be open on your trial, or included at least, it's not solely connected with melanoma. But from your point of view, though, why target PRAME, and what makes it particularly attractive to a TCR cell therapy developer?
Yeah, happy to discuss this. So PRAME is what is a so-called cancer testis antigen. It's expressed in a limited number of healthy tissues, such as testis, as the name says, but it's frequently expressed across a large range of cancer types. And as you said, initially it was identified as being expressed by melanoma cells, and it also has been associated with poor prognosis across a couple of cancer types. From a function perspective, we don't know all about PRAME. It's known to be a pro-tumorigenic factor, but really what the molecular function is, And in tumors, is not yet really fully known. So we rather use it as a marker that is, frequently present in tumor cells. But we, at TNF, we've really done a lot of studies to understand PRAME expression very well, because it's a late program. We really wanted to get a deep understanding of that protein in tumors. And everything that we've seen is really encouraging to position it as a really great target for TCR-T therapy. We see it expressed across a very large range of tumors, and particularly there's a number of indications, most of them which are included in our ATLAS study now, where the prevalence of PRAME is also very high. To give you an example, in ovarian cancer, almost all tumors are actually positive for PRAME, so it's really frequent. And what we are seeing, interestingly, on a biology perspective, is that this prevalence also correlates with very high expression level and with homogeneity of expression in our focus indications. And so these homogeneity and expression level have both been associated with better therapeutic outcomes. So that's really important for a target antigen. And another interesting factor for design is that PRAME is reactivated in tumors by a DNA methylation event. So in some time during the tumor development, the gene region where PRAME is encoded is getting demethylated, and then that's when PRAME expression basically starts. And the interesting piece is that this seems to be a mostly irreversible process. So it also means that once PRAME is turned on in a tumor, it doesn't go away. And so what we can see in our data is that it's very stable across disease progression. When we follow patients during different lines of treatment or, from diagnosis to later stages, they remain positive, and PRAME frequency remains very stable. And we see the same stability of PRAME expression also between primary tumors and metastases. So what it means for us as a developer is that, once we have a PRAME positive biopsy from a patient, there's a high likelihood that the T cells that enter that patient therapeutically will find PRAME positive cells across the tumor and in the different metastases and, at the time point when the patient gets treated. So I think from a lot of different angles, it's a very interesting target for tumors. And at the same time, it's a very safe target. So all of our studies say it's very lowly expressed only in a very limited set of healthy tissues where we are not concerned around on-target toxicity.
All of which is absolutely fascinating, actually. I hadn't actually realized that level of detail was understood about its actual role itself, potentially in the tumorigenesis as well. It's really, really interesting. And you're obviously not alone either in targeting PRAME. Several other therapy developers are also in the space. I mean, Immatics is already in a phase 3 study in melanoma and has shown additional clinical data in earlier indications with a second generation product. So where does that leave you from a kind of a competitive point of view?
I would say that we actually have the good fortune that PRAME as a target is already heavily de-risked in patients, in particular TCR-T as a modality, in contrast to much of the earlier targets which actually led to on-target toxicity in patients. The data we've seen from the field so far really show that it can be safely targeted by PRAME TCR-T. And importantly, PRAME TCR-T has shown really astonishing results in late-stage cancer patients already. And as you mentioned, particularly in melanoma, but also in synovial sarcoma. And just to give you one example about these data, so patients with uveal melanoma, which is a form of melanoma of the eye, which is a much less common subtype of melanoma, and typically considered a cold tumor, where a lot of standard melanoma treatments do not have the desired effect. In this indication, there was an initial phase 1 with PRAME TCR-T, and that showed a 67% overall response rate in this patient population and a median progression-free survival of 8.5 months. And these data actually caused a lot of excitement around this year's ESMO conference. And if they can be confirmed actually in a larger cohort, I think this is a real game changer for these patients. But what we're also seeing is that the field has really faced the challenge that how we can translate these great data and initial indications to more broader indications that are maybe more harder to treat opposed to melanoma, so that are not niche indications like synovial sarcoma, which don't represent a commercially enough attractive market. So we're looking at lung or ovarian cancer. How about treating these? And we believe that the field here is currently really at a breakthrough, right? We are now seeing these next generation TCR-Ts entering the clinic, where the T cells are getting engineered with additional molecules beyond just the T cell receptor. And these will further boost the immune response. And what we've now seen is really, a first phase 1 data coming out from such a second generation PRAME TCR-T in broader solid tumor indications. And these data just presented at ESMO IO actually extremely promising and will be, really exciting to see how much more in that field with next generation TCR-Ts.
Yeah, and I guess that's where TK6302, the TNF therapeutics therapy, comes in. Now, you are obviously a couple of years behind some of your competitors. What gives you confidence that you can be competitive? What are its key differentiating features?
Yeah, let's talk a little about the TK6302, right? So at T9, what we're doing is we're calling that supercharging T cells, which essentially means engineering new functions into T cells that were particularly designed through synthetic biology to overcome specific challenges that T cells face in the tumor microenvironment, and particularly in the tumor microenvironment of how to treat tumors. Our current lead product, TK6302, what we're doing here is we're combining a high affinity brain TCR that comes from our own proprietary MIT platform, and we're combining it with a co-stimulatory CD8 co-receptor and a so-called switch receptor. And so these components are really interesting. So the synthetic co-stimulatory CD8 co-receptor, a really hard to pronounce word, it allows us to integrate also CD4 T helper cells in the anti-PRAME response. And these CD4 cells in turn are really thought to really orchestrate a more global immune response, right? So they are really important to attract other immune cells and really get the immune response against the tumor rolling. And at the same time, we've engineered in here co-stimulatory domains, which are activating the T cells more optimally in the tumor. So that's a very novel function that has not yet been tested clinically. And that second component, which is really a major differentiator also, is what we're calling a switch receptor. This is designed to block inhibitory signal that's coming from the solid tumor microenvironment. And namely, that inhibitory signal that we're blocking here is Fas ligand. We're flipping that inhibitory signal into an activation signal for the T cell instead. Yeah? So leveraging it in a positive way instead of letting it act in a negative way. And so, yeah, this product is really extremely novel. It's very differentiated, and that's how we hope to be competitive.
Yeah, okay. And obviously the data will be watched with interest, and obviously that will take time for the data to mature. But so far, how has it performed in a preclinical kind of settings?
Yeah, so preclinically, we definitely see a highly differentiated profile of these supercharged PRAME T cells. We see, for example, that they're very activated, that they maintain a more sustained anti-tumor functions, that the T cells do not exhaust over time. And we've even developed a very sophisticated preclinical model, which is a 3D tumor spheroid model. And that model really represents more naturally the actual challenges in the tumor microenvironment for T cells. And in this particular model, and these are data that we just recently also showed at SITC, we see that TK6302 really outperforms other brain targeting approaches when we benchmark that side by side. So yeah, we really hope that this preclinical data, which are very clear, will also now translate into deep and durable responses in these brain indications, in particular outside of melanoma, which have so far been just harder to treat.
Yeah, fair enough. And then manufacturing obviously is so important with all therapeutics, but particularly this complex category of therapeutics. And it's been stated time and time again that cell therapies of any kind, that the process is the product. A clinical batch will need to meet a very complex set of quality requirements, which will hopefully correlate with its expected performance before it can be released to a patient. And what are the key steps in your production process for TK6302? And where do you stand in the process development kind of piece?
Yeah, for manufacturing, we're using a very novel approach, which is non-viral gene editing, which gives us actually a lot of advantages over traditional viral vector approaches for ex vivo cell therapy. And the advantages are, one, that we can actually use a much larger DNA cargo, which we need because we need to, in addition to the TCR, integrate that co-receptor and the switch receptor that we talked about. The second advantage really is that while we're knocking in the new genes, we also simultaneously knock out the endogenous TCR. So that's a thing you can only do with gene editing and not with viral vectors. Knocking out of the endogenous TCR really allows you to boost much more the expression of the TCR, resist the safety. So there's a lot of benefits for a TCRT in particular to be able to do that.
Mm. And by the way, too, what is the non-viral approach? How are you actually transfecting the cells with the cargo?
Yeah, so we're using a CRISPR-based approach, and we're getting it into the cells through an electroporation.
Oh, yeah, yeah, yeah. And then in terms of getting to dose levels, et cetera, where are you at with scaling to the level that you need to scale?
Yeah, really, really important. So we've locked the process. So the process development is actually finalized. We've locked it, and we could really demonstrate that we have very consistent batches and being able to generate the highest target cell dose actually that we're planning to use in the ATLAS study. We've also demonstrated, and I think that's a key point, we've demonstrated that this process also works for patient material, because you usually do the development only on healthy donor material, which not the fitness T cells, just very different.
Yeah, I totally get that point, because obviously the patients will typically be immunocompromised or in some way they've been through multiple rounds of therapy. A healthy donor isn't the same thing at all. It's a very interesting point. Okay. And I presume you're working with the CDMO then to actually do the actual process work. Is that right, in the live setting?
Correct. Yes, we've now successfully transferred the process to a CDMO that will do the clinical batch manufacturing, and they can reproduce the data that we've generated in-house with regards to batch manufacturing success. And we're really happy to now move that forward into the clinic.
So it's really all systems go. And then on the commercial side, what's the strategy for 6302? Do you want to get a partner for this, or do you want to maybe commercialize it yourselves, or if you're seeking a deal, what would be the ideal timing for a deal?
Yeah, it's early, but it's not early enough to actually ask the question, right? So I think in the near term, we're really focused on demonstrating clinical proof of concept for TK632, which means that there needs to be a real added benefit for patients, right? And a really differentiated profile for it. And demonstrating that supercharging T cells can really crack the code for how to treat solid tumors. That's really what we're aiming to do. That being said, I think for sure, right? We've looked at long-term plans. We have set up a CMC development plan that brings down costs over time and really think about how the commercial journey could look like, right? Over time. So that's definitely already strategies in place for that. And with all that work, I would say we feel really good about the business model for an autologous cell therapy for targeting brain. And you asked about partner. I think the right partner can be a real benefit for the program, both with regards to upscaling, patient access. So I think you can just leverage a different set of resources there. So for sure, we're open to partnering, and we also continue to build relationships with larger companies that are active in the space. But I would say right now, first and foremost, for us, it's about showing what we can make a difference for patients.
Yeah. And when you do that, everything else follows, really. Yeah, I would take that point. So I wish you and your colleagues all the best in these endeavors. And thanks for joining us this week, Elisa.
Yeah, thanks for having me. And as our team would say, better stay supercharged.
And thanks everyone for listening to this episode of BioBiz Buzz. We welcome your feedback as usual, comments or thoughts on this or other episodes. So if you want to get in touch, please log on to biobizbuzz.com or comment on LinkedIn, Blue Sky, X, or Facebook. And please continue listening on Apple Podcasts, Spotify, YouTube, or wherever else you may find your podcasts. In the meantime, goodbye for now.